Let's consider the pathophysiology of sickle cell anemia. But first, let's take a look at the hemoglobin, oxygen-carrying protein in red blood cells. Here's a graphic that shows the healthy hemoglobin protein, hemoglobin A. It's clearly made up of 2 alpha subunits in red and 2 beta subunits in green. And each of these subunits has associated with it a oxygen carrying heme group. And hemoglobin is actually a pretty cool protein. The subunits of the protein are able to communicate with one another and tell each other when they've bound oxygen. It's an allosteric protein that's very dynamic. So here's the hemoglobin protein here. Remember that individuals with sickle cell anemia have a defective hemoglobin. It's called hemoglobin S. So let's take a look at how hemoglobin S is produced. Here is the sequence of nucleotides in the DNA that give rise to the sequence of amino acids in the protein. You'll notice at position 6 in normal hemoglobin, this is the beta chain for hemoglobin A. And at position 6, actually it's on the surface of the protein, there is the amino acid glutamate. And so glutamate is present at position 6 in hemoglobin A. Take a look at glutamate for a moment. It's present on the surface of the protein. This kind of makes a lot of sense because see how the glutamate residue is very polar. It's very charged and it's hydrophilic. It's a hydrophilic residue. Now, with the missense mutation that occurs in sickle cell anemia, there is a change. There is a change in the DNA such that the GAG becomes a GTG. And because of that, we no longer get a glutamate in the protein. Rather, in the beta chain of sickle cell hemoglobin at position 6, there is a valine residue. So this valine residue occurs because of this missense point mutation. Point mutation is where there's just one change in one base, one nucleotide. But take a look at valine, for example. This is the amino acid valine down here. We clearly see that it is non-polar. It's non-polar, whereas the glutamate was charged and it was very polar. So these two amino acids are very different. The valine residue is very hydrophobic. And this change in just one amino acid, because it's such a significant change, because the amino acids are just so different, it causes quite significant changes to the hemoglobin protein. The change, just this one amino acid change to the hemoglobin protein, this one substitution produces an aggregate. It causes the hemoglobin to adopt a completely different structure. See how the regular hemoglobin, the normal hemoglobin molecule, exists as a tetramer. A tetramer. That means it's got the, let me just correct this. It contains these 4 different subunits, 2 alphas and 2 betas. But with this single amino acid change, do you see how now the hemoglobin is adopting more of a fibrous structure. It's adopting more of a fibrous structure, and these solid, insoluble fibrous arrays tend to occur particularly upon deoxygenation. So when oxygen is given up, when the hemoglobin gives off its oxygen, these fibrous arrays occur in the deoxygenated state. Deoxygenated state. And so this causes the red blood cells, this change in the structure of hemoglobin causes the red blood cells to adopt this sickled or crescent shaped configuration. These crescent shaped red blood cells, they tend to die more quickly. They have a half-life in the circulation of around about 10 days, about 10 to 20 days. So because of that, the circulating red blood cell levels in individuals suffering from sickle cell anemia, they tend to decline. The bone marrow can't keep up with making new red blood cells fast enough to replace the dying cells. Therefore, the blood content of erythrocytes is lower. And this is what leads to the chronic anemia that I mentioned previously. So this is a serious manifestation of the condition.